Welcome to this tutorial on oxidative stress, brought to you by Medical Biochemistry Power Capsule series. It is recommended that you also watch the tutorial 7b video, which is available in this series. This video covers the following CBME curriculum topic: describe the role of oxidative stress in the pathogenesis of conditions, such as cancer, complications of diabetes mellitus and atherosclerosis. Oxidative stress plays a significant role in the pathogenesis of various conditions, including cancer, complications of diabetes mellitus, and atherosclerosis. Oxidative stress can lead to DNA damage, which is a critical factor in the development of cancer. Oxidative stress can also activate signaling pathways that promote cell proliferation and survival, which are characteristics of cancer cells. Diabetes is associated with increased oxidative stress due to various factors including hypoglycemia, insulin resistance, and mitochondrial dysfunction. Oxidative stress plays a crucial role in the development and progression of atherosclerosis, which is a major underlying cause of cardiovascular diseases like heart attacks and strokes. Oxidative stress contributes to the initiation, promotion, and progression of cancer through various mechanisms. Oxidative stress contributes to cancer development by causing DNA damage, activating pro-survival signaling pathways, promoting immune evasion, and facilitating metastasis. Targeting oxidative stress and its associated signaling pathways may offer promising therapeutic strategies for cancer prevention and treatment. Given here is a list of mechanisms by which oxidative stress may cause cancer: DNA damage and mutagenesis, activation of signaling pathways, oncogene modulation, tumor suppressor genes modulation, immune evasion and metastasis, and epithelial mesenchymal transition. And we have a mnemonic for you, which will help you to remember the above list. DNA damage and mutagenesis. Reactive oxygen species generated during oxidative stress can directly damage DNA by causing modifications to its structure, such as base modifications, DNA strand breaks, and DNA protein cross-links. DNA damage induced by reactive oxygen species can result in mutations in critical genes involved in cell cycle regulation, DNA repair, and apoptosis. These mutations can lead to the uncontrolled proliferation of cells. Reactive oxygen species-induced DNA damage can also contribute to genomic instability, a feature of many cancers, by impairing the fidelity of DNA replication and repair mechanisms. Activation of signaling pathways. Oxidative stress can activate various signaling pathways that promote cell survival, proliferation, and tumor growth. Reactive oxygen species-mediated activation of receptor tyrosine kinases, such as epidermal growth factor receptor and insulin-like growth factor receptor, can stimulate downstream signaling cascades involved in cell proliferation and survival. Reactive oxygen species can also activate transcription factors, such as nuclear factor kappa B and hypoxia-inducible factor 1 alpha, which regulate the expression of genes involved in inflammation, angiogenesis, and metastasis. Oxidative stress can modulate the activity of oncogenes and tumor suppressor genes through epigenetic modifications, altering gene expression patterns that promote tumorigenesis. Immune evasion and metastasis. Oxidative stress can influence the tumor microenvironment by promoting immune evasion and facilitating metastasis. ROS can suppress the activity of immune cells, such as T cells and natural killer NK cells, impairing their ability to recognize and eliminate cancer cells. Oxidative stress-induced inflammation can promote the recruitment of immune-suppressive cells, such as myeloid-derived suppressor cells and regulatory T cells, creating an immunosuppressive milieu that facilitates tumor growth and metastasis. Reactive oxygen species can also induce epithelial mesenchymal transition, a process by which cancer cells acquire invasive and metastatic properties, through the activation of signaling pathways involved in cell motility and extracellular matrix remodeling during EMT. Epithelial cells acquire mesenchymal properties, promoting metastasis and therapeutic resistance. Through EMT, cancer cells gain enhanced migratory and invasive capabilities. Oxidative stress plays a central role in the pathogenesis of complications associated with diabetes mellitus by inducing tissue damage, inflammation, and impaired cellular function. Oxidative stress contributes to diabetic complications such as nephropathy, retinopathy, neuropathy, and cardiovascular disease by damaging cells and tissues, impairing their function, and promoting inflammation and fibrosis. Here is a list of mechanisms involved: mitochondrial dysfunction, hyperglycemia-induced reactive oxygen species production, activation of pro-inflammatory pathways, impaired antioxidant defense mechanisms, and formation of advanced glycation end products. And here's another mnemonic, which will help you to remember these points. Mitochondrial dysfunction, hyperglycemia and insulin resistance can impair mitochondrial function in cells, leading to increased mitochondrial reactive oxygen species production. Mitochondria are major sources of reactive oxygen species within cells, particularly during conditions of metabolic stress. Dysfunctional mitochondria produce excess reactive oxygen species as byproducts of oxidative phosphorylation, contributing to oxidative stress. Hyperglycemia-induced reactive oxygen species production. Hyperglycemia leads to increased production of reactive oxygen species through various pathways. One major pathway is the glucose autoxidation process, where glucose reacts with molecular oxygen to form superoxide radicals and hydrogen peroxide. Additionally, hyperglycemia can stimulate the production of reactive oxygen species via the activation of enzymes such as NADPH oxidase and xanthine oxidase, which generate superoxide radicals. The illustration depicted below will further clarify the points which have been described above. Activation of pro-inflammatory pathways. Oxidative stress in diabetes mellitus can activate various pro-inflammatory pathways, exacerbating tissue damage and contributing to the development of complications. Reactive oxygen species can activate nuclear factor kappa B, a transcription factor that regulates the expression of pro-inflammatory genes involved in cytokine production and immune cell recruitment. Chronic inflammation driven by oxidative stress plays a key role in the pathogenesis of diabetic complications such as nephropathy, retinopathy, neuropathy, and cardiovascular disease. Impaired antioxidant defense mechanisms. Diabetes mellitus is associated with impaired antioxidant defense mechanisms, further exacerbating oxidative stress. Chronic hyperglycemia can deplete antioxidant reserves and impair the activity of antioxidant enzymes such as superoxide dismutase, catalase, and glutathione peroxidase. Reduced antioxidant capacity allows reactive oxygen species to accumulate unchecked, leading to cellular damage and dysfunction. Formation of advanced glycation end products. Hyperglycemia can lead to the non-enzymatic glycation of proteins and lipids, resulting in the formation of advanced glycation end products. These substances contribute to oxidative stress by generating reactive oxygen species through receptor-mediated mechanisms, such as the interaction with the receptor for advanced glycation end products and activation of NADPH oxidase. Additionally, advanced glycation end products can cross-link proteins and alter their structure and function, leading to tissue damage and dysfunction. Oxidative stress plays a critical role in the pathogenesis of atherosclerosis by promoting LDL oxidation, endothelial dysfunction, inflammation, smooth muscle cell proliferation, and plaque vulnerability. Strategies aimed at reducing oxidative stress may hold therapeutic potential for preventing and treating atherosclerotic cardiovascular diseases. Here's a list of the probable mechanisms involved: induction of endothelial dysfunction, promotion of inflammatory responses, oxidative modification of LDL cholesterol, stimulation of smooth muscle cell proliferation and migration, and oxidative stress in plaque rupture and thrombosis. And yet another funny mnemonic, which you would definitely find useful. Induction of endothelial dysfunction. Oxidative stress contributes to endothelial dysfunction, a key early event in the development of atherosclerosis. Reactive oxygen species can impair endothelial nitric oxide synthase activity and reduce the bioavailability of nitric oxide, which is a crucial vasodilator and anti-inflammatory molecule. Reduced nitric oxide availability promotes vasoconstriction, platelet aggregation, and leukocyte adhesion to the endothelium, initiating the inflammatory response characteristic of atherosclerosis. Promotion of inflammatory responses. Oxidative stress triggers inflammatory signaling pathways within the arterial wall, promoting the recruitment of immune cells and the formation of atherosclerotic plaques, as in other cases. Here also reactive oxygen species can activate nuclear factor kappa B, a transcription factor that regulates the expression of pro-inflammatory genes involved in leukocyte adhesion molecule expression, cytokine production, and chemokine secretion. Inflammatory mediators, such as interleukin-6, tumor necrosis factor alpha, and monocyte chemoattractant protein 1, contribute to the recruitment of monocytes or macrophages into the vessel wall, where they engulf oxidized LDL and transform into foam cells. Oxidative modification of LDL cholesterol. Low-density lipoprotein cholesterol is susceptible to oxidative modification by reactive oxygen species within the arterial wall. Oxidized low-density lipoprotein is formed when reactive oxygen species, such as superoxide radicals and hydrogen peroxide, oxidize the lipid components of LDL particles. Oxidized LDL has altered biochemical properties compared to native LDL, making it more prone to uptake by macrophages, leading to the formation of foam cells, a characteristic feature of early atherosclerotic lesions. Stimulation of smooth muscle cell proliferation and migration. Oxidative stress promotes the proliferation and migration of vascular smooth muscle cells, contributing to the progression of atherosclerosis. Reactive oxygen species can activate growth factor signaling pathways, such as platelet-derived growth factor and transforming growth factor beta, which stimulate vascular smooth muscle cells proliferation and migration from the media to the intima. In the intima, proliferating vascular smooth muscle cells contribute to the formation of the fibrous cap that covers the atherosclerotic plaque. But they can also exacerbate plaque instability if their proliferation is uncontrolled. Oxidative stress in plaque rupture and thrombosis. Advanced atherosclerotic plaques with a large lipid-rich core and a thin fibrous cap are prone to rupture, leading to acute thrombotic events such as myocardial infarction and stroke. Oxidative stress within the plaque contributes to plaque vulnerability by promoting the degradation of the extracellular matrix and weakening the fibrous cap. Additionally, reactive oxygen species can activate platelets and promote thrombus formation by enhancing platelet aggregation and adhesion to the exposed subendothelial matrix.
Thanks for watching this video. Our next tutorial in the Medical Biochemistry Power Capsule series is titled Diet and Nutrition. The tutorials presented in the MBPC series are not intended to be a substitute for your textbooks. However, we feel that these tutorials can prove to be very valuable as complementary educational aids and for purpose of rapid revision prior to exams. If you have enjoyed this video, don't forget to hit the like button, subscribe for more exciting content, and share with your friends. And do keep watching our channel to remain ahead in the game.